Okay, so yeah, so obviously there's a group of us that are working on winter barley. That's what I'm going to talk about today is how can we bring winter barley into the mix of crops that are part of this Forever Green initiative. So there's a bunch of folks that are working on this. Some of them you may have heard of make presentations in the past from Aaron's group about looking at sort of the soybean side of a winter barley soybean cropping system in Walid, and folks in his lab talking about some of the work they're doing in looking at sort of the biophysics of freezing tolerance in winter barley and some really interesting results there. Stephenson's lab has been very diligently screening lots of germplasm to look for winter hardiness, sources of low temperature tolerance or winter hardiness that we can use in our breeding program. And then the folks in my group have been working a lot on various breeding and genetics aspects of that would feed into this winter barley system. And then we have a network of collaborators that we're working with too. I coordinate a national winter barley malting trial. It has some years up to 28 different locations across the US for the last 11 years. And so that's been a really fruitful network of folks to work with as well. And then we're getting funding from a variety of different sources to support this work. But today I want to just focus on two things, or a couple of things. So we'll break this up into three parts. I'll start out and I'll just say a few words about why we're going to talk about winter barley. I'll give you a little bit of an update, part of an update of what we're doing in the breeding program. And then Ahmed Salam will talk, who's a postdoc in Brian Stephenson's lab, will talk about the work that he's been doing in looking at the genetics of low temperature tolerance, which is the biggest battle we have to fight in terms of the work that we're doing with winter barley, is trying to improve winter survival. And then the last part, I'll switch back and I'll talk about the last part. And the last part is going to be really a proposal for an experiment that looks at a winter barley soybean cropping system. And since sort of agronomic cropping system experiments are outside of my realm of expertise, I'm going to really draw from a lot of experts I know that are in this audience to help us think through this experiment, because I roughed out a proposal. The proposal was funded, so we have some money from Minnesota Department of Ag as one of the objectives of this proposal. But it's not going to start until the fall. So I thought, rather than waiting 3 years when I present the data from that experiment and then you guys rip it apart because it was all designed poorly, that maybe we could talk about it now, make sure that we got some of the design features figured out and can anticipate maybe some of the issues, so that going into the fall, we can really have a rock solid experiment design. So that part of the presentation, I'll be particularly looking for feedback from the group, but as always, I think anybody can jump in and interrupt and stop and ask questions as we go. Okay, so some basics about winter barley. Obviously, as a winter annual, we're interested in it for a number of different reasons. This is just a picture that tries to illustrate some of that in terms of the way that this is taken on the same day, a spring planted and fall planted. Obviously, you've got more biomass there. You've got probably the potential to reduce erosion. All of the things that we think about when we think about winter annuals in a cropping system. And so that's obviously a big driver of why I first started getting interested in winter barley. Another thing is that, in the literature, a lot of places with winter cereals, there is a potential to have higher yields in winter barley. And that's illustrated in this slide. But before I go into the data in the slide, I just want to make sure I explain what I mean by winter barley. When I say winter barley in our breeding program, what I'm talking about is actually facultative barley. So facultative barleys are barleys that do not require a vernalization period. So they don't require a cold period in the winter in order to flower. So you can either spring plant them or fall plant them. And there's quite a bit of evidence in barley that that requirement for vernalization is not needed for winter hardiness. So you can have facultative barleys that are just as winter hardy as true winters. And obviously facultative barleys, because you don't require that cold period for vernalization, offer lots of advantages from a breeder's perspective. I can do more generations per year. I can cross with my spring lines. It's very convenient to work with a facultative barley. From a producer's point of view, the ability to spring plant or fall plant the same variety could be quite useful. From a seed producer's point of view, like somebody, like I talked to folks at Albert Lee Seeds, they have trouble keeping in stock winter barley sometimes, because if they got a really bad winter and there's no production, then their seed stocks are very low. Well, with a facultative barley, you could basically be growing your seed stock out over the summer, harvest that, and then and there's no risk of winter, kill, and then produce that seed for growers. So facultative barley is what we really do in our program, and we select for facultatives quite early in the program. So now to this slide, what you're seeing here is the results of a trial last year where we had a fall planted and a spring planted trial side by side in St. Paul with the exact same entries, because they were all facultative barleys. And you can see that there's a fairly substantial yield difference. And this was a pretty striking yield difference. This is larger than what a lot of people report in the literature, but most comparisons that look at spring and fall planted, you will see a substantial increase in yield, because the barley is able to take advantage of all of that photosynthesis and sunshine that's happening early in the spring season before a typical spring crop is planted. And it also avoids the heat of the summer during flowering and grain fill. So we think increased yield is a potential. Of course, that requires that it survives the winter, but assuming it survives the winter, that's a possibility. Another thing that I think is really important is that winter barley can be harvested about 3 weeks earlier than spring barley. So that's a substantial difference in when harvest would occur. Again, this is the same set of lines, spring planted and fall planted, in this side-by-side trial. And this is important because it would help, in terms of just farmer operations. You're spreading out those operations over the summer. We think this can be very important in avoiding some diseases that typically don't come on until a little bit later in the growing season. But if we get an earlier growing season, we can avoid some of those diseases, like some of the rusts. And then if we can shorten the season enough, it offers an opportunity for the potential for some sort of double cropping system, which is what we'll talk about towards the end of today's session. And then the last reason I'll throw up here that we're interested in winter barley is that the current spring barley production area has moved steadily north and west, away from a lot of the malting and brewing infrastructure. And basically it's concentrated in the region where there's the least amount of brewing going on. So if you look at the map on the left, it's a little bit outdated, but it kind of shows you where a lot of the breweries have sprouted up across the US. And there's a sort of a disconnect between where the barley is produced and where those brewers exist. And the other thing is that we have in the state of Minnesota, the largest single site malt plant in the world with raw malting. And so they have a huge demand for barley that they produce malt from and sell internationally. So they're very interested in shortening the distance that they have to transport barley to their site for malting, and winter barley might offer that possibility. So if it's such a great deal, what's the problem? Why aren't we growing lots of winter barley? And the obvious reason is that winter survival is not a guarantee in every year that we grow barley. And so this is a year where we had sort of differential survival. So you can see, lines that look like they survived very well and lines that didn't survive well at all. This is my favorite of all the different traits and phenotypes that I score in breeding programs. This is my favorite trait. It's the easiest one to score. And so our big challenge is working on winter survival. And Ahmed will talk in a couple of minutes about the work that we're doing in trying to really optimize the genetics for winter survival in barley. So the problem with winter survival is that you can be breeding, and this is a breeding scheme on the left. You can see sort of a typical scheme where we're making crosses with parents, advancing lines through the F1, F2, F3, F4, F5 stage, and then into yield trials, and then into industry scale brewing trials to get a variety in about maybe 9 years if we're lucky. But if somewhere along that pipeline you get a complete winter kill year, you lose everything. And so Brian Stephenson actually attributes this quote of breed until you run out of seed to Jim Anderson when he was a winter wheat breeder. So I don't know. I actually never asked Jim if that was, if he remembers actually saying that. I don't know if he's on. Can't tell if he's on today's or not, but you can jump in and tell us if we're misappropriating this quote to you. But this is what can happen. So this is a field, I think this was in the 2016-17 growing season, where virtually everything in the field was wiped out. And these are F3 hedgerows that would have been growing in this field. I wrote in here 15% of the F3 hedgerows had like one surviving plant. So most of those rows really had like one surviving plant. But there were some plots, and we did replicates and some F3s where we actually had multiple plants surviving. And so we did actually dig them up, put them in pots, bring them into the greenhouse, and we made some crosses with them. And some of those crosses actually have stayed on our program and are in our second year field trials right now. So this is a challenge though, because you can't just wipe out your breeding program every 4 or 5 years when you have a big serious winter kill event. So one of the things we tried to do is to develop this, what I call semi-controlled low temperature screening. So we're trying to produce winter without all of the winter. And so you can see there's a little hoop house here to the left of our greenhouse where we can we can move plants into that hoop house during the winter. We can very roughly control the temperature in that hoop house to kind of approximate some kind of winter screening in a way that we will hopefully not lose everything, but impose enough and do this on a large enough scale. So this is a larger scale than what we could do in a controlled programmable chamber that is under high demand. And when we did this last year, I think I showed this actually last year, we had very rough control. So we were working on getting ways of control. And then we had a power outage on the day when the temperature dropped below 0. And everything just crashed. And the result is that everything died pretty, there were 1 or 2 little green plants, and I don't know that it actually produced seed. So we did that experiment again this year with a little better success. So we were able to control the temperature a little bit better during this kind of this acclimation phase. And then we turned all the heaters off, and we just kind of let things go cool. We stopped it by about 10 degrees instead of getting it down below 0, and then let it recover. And this seems a little closer to what we would like to get. So we have some survivors in here. We probably killed off 80% and have about 20% survivors. So we'll see what happens with these guys as we advance them through the program. So this is just one method. We're trying to sort of mitigate the possibility, but our strategy right now is really to have several different methods of advancing in this very early generation stage. We'd like to do as much selection as possible. So on the left in the field, I'm showing you kind of what we had done, which is just to grow these things in the field. And if they survive, they survive. If they all die, then well, we're stuck. The hoop house I just showed you gives us an opportunity. We can actually grow F2s in the fall, screen our F3s in the winter in the hoop house, and then grow out the F3, 4 generation in the field, and then advance those into trials. So it allows us to actually do a little bit of single seed descent while we do some screening. And then our backup is just to not impose any cold on them at all, just make sure everything gets through the program in case these other methods essentially kill everything. So we're trying to make sure that we have something moving through the program every year, and we'll utilize whichever of these systems gives us the best opportunity for screening for winter hardiness. So where we sit right now is that we have we had a 6-row winter program for quite a while, and that things are moving along in there, but the malting and brewing industry has exhibited or has expressed a strong interest in only 2-row barley now. A couple of years ago, they made that decision. So we had already had a 2-row program going, but the status of our facultative 2-row program is that we have lines that are currently in our second year of yield trial testing right now. So this year we'll be pulling things out of that second year of testing, and hopefully then we'll be able to start putting them into regional trials and our AMBA, which is American Malt and Barley Association industry pilot malting trials to see if they have a potential for variety release. So that's kind of where we are at our program right now. And I think what we'll do now is I'll stop sharing my screen and I'll give Ahmed a moment to get his screen up. But I'll pause there for a minute in case folks have questions so far.
Kevin, what's the rationale to go to 2-row barley in the industry?
Well, so the industry has historically used a mixture of 2-row and 6-row barley. The 6-row came mostly from the Midwest. The 2-row came mostly from the West. More of the production is being grown at the West, so they've kind of been forced to go more 2-row anyway. I mean, that's in there, and they've built more infrastructure there. And then the craft brewers are all malt brewers are very much interested in 2-rows because most 2-row varieties are a better malting profile for all malt brewing. So there's been this general trend for this increase in 2-row, interest in 2-row. I think that even the big brewers just decided we would rather figure out how to just use all 2-row than how to keep, a 6-row in the mix, even though 6-rows still actually do better in the Midwest in terms of yield and disease resistance and straw strength. And there are a lot of producers in Minnesota and North Dakota that are quite frustrated with having to switch to 2-row. And what we're actually going to see is a drop in acreage, pretty substantial drop in acreage, even more in the Midwest and Minnesota and North Dakota this year, I'm pretty sure.
Until you have lines that are better adapted to Minnesota, is that what you're saying?
Yes. So I wish the industry would have waited until we had some spring 2-row types that would have been more attractive to growers before they sort of unilaterally made the switch. But that's how it happened.
Kevin, Carmen here.
Yeah.
Any work on planting date for the fall when you plant in the fall, and then any research on snow cover impact?
So in terms of planting date, yeah, so we had a former student, Becky Zong, did some work on planting date. And we found that later planting dates actually worked quite well. So like roughly October 1 planting date was a pretty good target. If we planted earlier than that, we sometimes saw issues with disease, like yellow dwarf or mildew or other things sort of, developing into the fall, and that hurt them in the spring. But we saw barley that was, if we had good snow cover and we had survival that year, even 2 leaves, into the fall survived quite well. So they don't need to be that far along for survival.
Okay.
And then the other question was snow cover is, we've just done sort of anecdotal. We've collected data on our own trials on snow cover. There's obviously a strong effect of snow cover. If you just look anecdotally at the data we have, I think if you have 2 or 3 inches of snow on the ground, it can get as cold as you want and the barley will probably survive. But the years where we have the real problems are when there's no snow and then we get a dry front move through and it drops down to like 0 or below 0 without any snow.
Well, that planting date is exciting because that could probably put us into planting barley after some soybean harvest.
Yes, and that's exactly the kind of thing that we're thinking about with double cropping. So yeah.
Great.
All right, well, I will turn it over to Ahmed and he can tell you about some of the genetics work that we're doing with winter survival.
Thank you, Kevin. Does everyone hear me and see the slides?
Yes.
Yes.
Sounds good. So I will talk about the efforts that we did in terms of the genetics for winter survival in barley. Of course, there is a need, as Kevin mentioned previously. We need to increase the genetic diversity for low temperature tolerance in barley for winter barley production in Minnesota and also northern states as well. We thought about utilizing the winter hardiness in the Vavilov barley collection. So we obtained a representative sample of 2,254. It was in the beginning, we were thinking about disease evaluation. Brian Stevenson brought those Vavilov diverse Vavilov barley accessions into Minnesota. And then Brian thought about evaluating them for winter survival, since many of those accessions came from cold countries, including Russia. So we did the first evaluation, the Vavilov collection for the 2,254 accessions in St. Paul, Minnesota in 2014-2015 growing season. From them, 268 accessions, about 12.1% survived. The severe winter conditions at this year were 99% of the breeding lines died off. Of course, these led to the current efforts for exploiting genetic diversity for low temperature tolerance or winter survival in barley. The 268 accessions originated from 35 different countries from 4 continents, so great level of genetic diversity. But about 107 of them, about 40%, originated from Russia, but still substantial number of accessions came from cold challenged countries, including Ukraine and Bulgaria. Subsequently, we evaluated the 268 accessions in 11 trials in 7 different states. From the west of the US, Idaho, Montana, Nebraska. In Minnesota, we put it in 3 locations, in Crookston, Minnesota, St. Paul, and Rosemont. We also put it in Alma, Wisconsin, Worcester, Ohio, and in New York at Ithaca. We included several chick controls, including Standard, which is a spring type. McGregor is a winter hardy barley, that's winter type. Dicto is one of the most winter hardy facultative barley types. We also included the winter types Charles and Maha, and we also included North Star wheat, which is one of the most winter tolerant small grains, and Raymond as well, which is a winter hardy rye. And we measured winter survival in all the locations as percentage of the plants survived after the winter. This picture was taken from Rosemont, Minnesota last year in April 2020. So you can see that Standard totally to the left died off. McGregor and Dicto survived at higher rates. And also we can see to the right 4 barley accessions from the Vavilov collection. Their survival is comparable to Dicto and McGregor. This is a picture for the whole Rosemont nursery. You can see that we had great survival at this cold conditions. After the cold conditions, we had almost all the plants of the 268 accessions survived. Of course, we managed to obtain valuable winter survival data from 7 different trials, because in Crookston, Minnesota, nothing survived, and we had some issues with the other locations, but at the end we had 7 strong data sets for winter survival across for the entire 268 accessions. This figure displays the distribution, and this box plots distribution of winter survival in the 7 trials in St. Paul, Minnesota, Aberdeen, Idaho, Alma, Wisconsin, Bozeman, Montana, Ithaca, New York, Meade, Nebraska, and Rosemont, Minnesota. And they have also included 4 checks: Standard, Charles, Dicto, and McGregor with different colors. And their names, first letter was included on the plots as well. So you can see that Standard almost died off in almost all trials, except at Bozeman, Montana, because of the depth of the snow coverage that they have at Montana. You can see that we have great distribution for winter survival among the 268 accessions. You can see that clearly in Aberdeen, Idaho, Alma, Wisconsin, Meade, Nebraska, and in Rosemont, Minnesota. We have several accessions that performed better than then the winter hardy checks, McGregor and Dicto and Charles across all the locations. We have from the 268 accessions, 78, best 78 that each had 50% or greater survival in each one of the trials. So we selected these 78 accessions and we found out that they were originated from 34 different countries, but most of those accessions came from former states of the USSR. Those 78 accessions were further evaluated in 3 trials at Montana, Nebraska, and at New York. This is the distribution for the 78 accessions at Mead, Nebraska. These trials were completed and scored by Dr. Stephen Benziger. We have also Moccasin, Montana, and at Ithaca, New York. I think this done with Dr. Mark Sorrells. So we can see that, and we have 2 common checks among all the trials, Charles and Decto. We can see that we have several accessions that perform better than Charles and Decto. This is very promising. I mentioned before that we have 2 checks common, the Decto and Charles. So I use those checks to correct for variance in trial means between the 7 trials that were done before and for the entire population and the re-evaluation for the 78. And the distribution of both those populations or the evaluations shown for all the veer collection in the light red color with the best 78 in the light blue color. Anyway, we can see that there is higher performance for winter survival for the selected 78 compared with the 267. So significant difference, 86.9% compared with 64%. We grow also the entire veer collection, the 267, the veer LTT panel in the summer, in the spring to determine the growth habits, facultative versus winter. So out of the 78 accessions, the best accessions, 42 of them were with facultative growth habits and 37 are with winter growth habits. And their means are equal. So this shows the expectations that we have, great expectations we could have with the facultative growth habits. Barley, as Kevin talked about this previously, it provides flexibility for both the growers and for the breeders. This figure display or table display the phenotypic performance for the best 10 individuals or accessions out of the 78 and sorted from high to low. However, they are not significantly different from each other. Number 1, I think, was Veer 2251. This is from Germany. We have also here Veer 1396. I'm going to talk about this later. It's from Russia. It shows great winter survival. We genotyped the population, and this is the genetic relationship among the accessions using molecular markers. The diagonal numbers here, these are lines by themselves, and the off diagonal are the lines with each other. So you can see that those are diverse low numbers. That means these kind of diverse accessions, except for those 2 accessions from Bulgaria. They are almost identical. So from these 10 performing accessions, we selected 9. We dropped one of those 2 Bulgarian accessions, and we selected 9 accessions, and many of them were used as parents for the barley breeding program. We genotyped the whole population, the 267 accessions with the 9K. I select array SNP chip, and we performed genome-wide association mapping to identify genes responsible for low temperature tolerance. This is called Manhattan plot, and we have 7 barley chromosomes, and physical positions are in the x-axis here, and the power of QTL detection or gene detection here on the y-axis. The dashed red line is for the threshold for the gene detection. Any significant hit over the bar, the red bar indicating a gene responsible for winter survival in barley. You can see that we have genes or QTLs across all the chromosomes except for chromosome 2H. One of those genes resides on the FRH2, which is very important frost-resistant gene that was identified previously. Our study is not the first study that performs genome-wide association mapping for winter survival. There is another study that was published previously, just recently. It's called the CAP LTT panel. This population is composed of many accessions from around the world, about 800, more than 800 accessions. The population also was genotyped using the same marker set that we have in the Vavilov collection. And also they performed genome-wide association mapping, the same method that we use in our study. And they detected the exact, from this is the table for the QTL mapping, and they identified the exact same marker or marker trait association at FRH2 that we identified. So this is indication that we managed both studies to validate each other in terms of the identification of the genes. They also identified another important genes for FRH3 in chromosome 1 and of FRH1 in chromosome 5. By the comparison of the results of the Vavilov collection and other studies, we had total of 7 novel associations that are highly significant on all barley chromosomes. This shows the potential of this population to improve winter survival in barley. We thought also about combining the results from the CAP LTT panel and the VIR LTT panel to increase the genetic diversity and exploit it. Of course, these are independent studies. They were not evaluated together. So we received all the phenotypic data for winter survival that they did. And I found out that we have 3 common checks between our trial for the VIR LTT panel and the CAP LTT panel. These 3 checks are Charles, Dick2, and McGregor. Therefore, I used those 3 checks to correct for variance in trial means. So we'll be able to combine the results of those studies together. I coupled this with the use of haplotypes. And this is how I generated haplotypes for both panels. After sorting markers based on the physical position for each chromosome from low to high, I generated haplotype blocks of fixed number of 3 adjacent markers. This kind of cartoon describes how I generated those haplotypes for each one of the 7 barley chromosomes in both panels. And I used those haplotypes and the phenotypic data that we received after we corrected them for variance in trials means to collect what's known as, to calculate what is known as haplotype additive effect. As I mentioned previously, both studies identified the frost-resistant gene FRH2. And this is in the Vavilov collection. We have here 3 markers in this haplotype. The most significant marker here is in the middle. And these are the nucleotides at each one at those markers. And then this is the haplotype, final haplotype. So TAG for the first haplotype, GGC for the second haplotype, and GAG for the third one. Those are the frequencies for each one of those haplotypes. And then these are the additive effects. We found out the GAG haplotype here that happened in 81% of the population has positive additive effect. In the CAP LTT panel, at the same gene FRH2, we have also the same set of markers that were used in the study. In this case, actually, we have 4 haplotypes because this is more diverse panel. It's more than 800 accessions, more larger populations who expect to have more genetic and allelic diversity. We found also that the same haplotype that resulted in the highest positive effect is also GAG, so that across these 3 markers. This is indication that we are on the right track and we are moving into the right direction of combining the results of both studies together. I performed the haplotype analysis in both panels for all marker trait associations, the 12 associations on the Vavilov collection and the 18 on the CAP LTT panel. And I managed to obtain the favorable haplotype allele for each one of the individuals, for each one of the genes. So we'll be able to identify for each one of the haplotypes, so identify the individuals carrying favorable haplotype alleles. So as I showed previously, FRH2, we have the GAG across 3 markers. This is a summary of the favorable haplotype alleles in the top 9 individuals that I talked about previously. These are the favorable haplotype alleles in blue color, and then indicating here if the individual carrying the favorable allele or not. You can see like the first accession from Germany here, the VIR 20-2051 has seven favorable haplotype alleles. VIR 1397 from Russia, 1376 from Russia has all the favorable haplotype alleles at all the marker trait associations that we identified. I did the same thing also for the CAP LTT panel. And our ultimate goal is to develop a multi-parent advanced generation intra-cross population or a magic population. We want to integrate the genetic diversity and favorable alleles from both panels into a single population. And the magic population could be created by intercrossing 8 parents that are very diverse, and they carry the highest number of favorable alleles. We perform 2-way crosses. First, we generate the 4-way crosses. We generate that first, do the 2-way crosses, and then we perform the 4-way crosses, and at the end, the 8-way crosses to generate 8-way F1 seeds that could be selfed for several generations to generate a large population. There are several advantages of using the magic population, including, of course, the richness of allelic diversity compared with biparental populations. Second, it will generate at the end an immortal population that could be phenotyped in several locations. If we decided to pick individuals only based on how many favorable alleles there are from both panels, we can see that from the Vavilov collection and from the CAP LTT panel, all the selected individuals will be either Winter 6-row or facultative 6-row, and all of them non-malting type. Of course, as we know, the breeding efforts are focused toward into facultative 2-row. That's why we had to make some selections on different selection or different criteria for making these crosses. We have total of 25 potential parents from for the barley magic population. That includes the best individuals from the CAP LTT panel, and also the best individuals from the Vavilov panel. And I have different colors on this table. They are colored based on growth habits, row type, and malting quality. For example, the blue accessions are facultative 2-row. The yellow accessions are winter 2-row. The green accessions are winter 6-row. And the light brown accessions are colored with facultative 6-row. So we decided to make specific selections by crossing the facultative 2-row with the winter 6-row. So the progeny will have at the end, since facultative is dominant over winter and the 2-row is dominant over 6-row, so the progeny most likely will be facultative 2-row. Of course, segregation happens, but the facultative 2-row should happen with higher frequency. Also, crossing the winter 2-row with the facultative 6-row, facultative 2-row with winter 2-row, and facultative 2-row with facultative 6-row. We were successful at the end. These crosses were done in the winter greenhouses 2020. We managed to obtain 100 different crosses, combinations. And also we genotyped the potential parents with molecular markers to estimate the genetic relationship between them. And we can see to the right that the 2-row malting types, facultative or winter, are clustered here to the right. And the 6-row non-malting, either winter or facultative, are clustered here to the left side of the tree. The Vavilov accessions with facultative 6-row growth habits, the facultative growth habits and 6-row are clustered together. And the winter 6-row from the Vavilov collections are also clustered here together. Finally, we selected the 8 parents for the magic population. And this is a summary for the favorable haplotype alleles that I talked about previously. And we have 3 colors in this plot. The brown for marker trait associations identified in the Vavilov collection. Cells with the light green colors are the marker trait associations identified in the CAP LTT panel. And with the blue colors are marker trait associations identified using both the Vavilov and the CAP LTT panel, in this case, FRH2 gene. We have we can see that we have several spectrum of favorable haplotype alleles for all those selected parents. We covered all the significant marker territory associations, all of them with those selection of the parents. In terms of the timeline for the development of the magic population, so we've completed the two-way crosses in the winter greenhouse of 2020. We just finished the four-way crosses in the current winter greenhouse of 2021. The eight-way crosses will be completed in the fall greenhouse next fall in 2021. Then the eight-way F1 seeds will be grown in the winter greenhouse of 2022, and then we'll start to advance those self-fed individuals, and hopefully be generating the populations by the year of 2023 to be evaluated in several locations. This is the last slide, and if you have any question, I'm open to answer any question.
When you cross a 2-row and a 6-row, what is the segregation for the 2 and 6 in the progeny?
Yes, this is expected, but we also are combining from other parents as well, that are other crosses as well, that are 2-row and 6-row. So that's why we plan to generate very large population, over 3,000. So we'll be able to recover facultative 2-row accessions with good winter survival.
What's the segregation ratio when you cross a 2 by a 6?
It's going to be 3 to 1.
3 to 1. Yeah.
At the F2. At the F2.
Yeah. Well, are you excited? You think this is going to? You think we're going to be able to, based on what your gut feeling, or you think we're going to be able to develop winter hardy 2-row?
I think that when I look at this table and we see that we covered everything was identified in both panels, and this is the CAP FTT panel is totally different study done by different groups. Kevin was involved in this because this population was also evaluated in Minnesota and also other countries around the world. So I think we covered, we have different spectrum. We covered every significant marker trait association. We have the favorable haplotype alleles for all the selected parents. So I'm very hopeful that we'll be able to identify at the end several individuals, facultative 2-row, with excellent winter survival.
So Ahmed, this is Gary. I have a question about that that particular Vavilov line that had all the haplotypes, all the favorable haplotypes.
Mm-hmm.
What sort of what's the phenotype of that line? Does it ever get killed?
It got killed in Crookston, Minnesota.
But it doesn't get killed in Nebraska or New York State or Idaho or Montana or Rosemont or St. Paul?
I have actually a table. Let me see. This is V13976 here. No, it shows always 60, 100, 60, 100, 60, 100, 90, 95, 100, 85. So it never, actually, it survives at a pretty high ratio.
So what's the percent survival that you need to get a good stand? So I assume that once you have like 60% survival, the plants are going to tiller more than 100% survival. So is 60% enough to get a reasonable stand and get enough spikes that are productive?
I think so. 50 to 60% or more. Yes, that will be sufficient. Yes, I think this will be sufficient, I believe. And Kevin can comment more on that.
Yeah, we did a little bit of work just kind of fitting some regressions, and I would say 70% was equal in yield to 100%. So something in that ballpark. Yeah, you don't need to get 100% survival.
Carmen here, what kind of seeding rate were you anticipating?
I mean, this was just the standard seeding rate, like two bushel an acre seeding rate. So, you know, but that's another variable, right? You could bump up the seeding rate if you think the risk is high enough.
Okay.
So I'm a little confused by the need for the magic population then. So if you've already got a line that is like super good, why don't you just use that line and start turning that into a malting quality variety versus trying to continue to map the cold tolerant or LTT QTL?
It's this is true, but also it's a winter 6-row non-malting. Plus, it misses also some important genes as well, like, because it didn't really have all the favorable alleles that were identified in the CAP LTT panel. I think this parent already in several is a parent to several lines. I think in Kevin PYT already this year. So it's a good parent, but I think it misses very important QTLs that were highly significant in the CAP LTT panel. So we need really to increase the frequency of those alleles in the individuals that we're generating. So be able to capture those, have this genetic variation to further improve winter survival in the cold Minnesota conditions.
So I have one other question. Thanks for that answer. So one other question. So there's lots of different alleles that are going to segregate in that magic population. What's the size of the population you're going to need to recover an individual that contains all of the favorable alleles for all the QTLs you identified in both of the 2 GWAS experiments?
I think the
It's
plan
going to be tens of thousands, I assume, right?
we actually our plan maybe like 3,000 breeding rails. So we can determine that later. But I think what we have so far, we have lots of crosses that were done in the current greenhouse. We'll be able to make all these types of process, further process. But we thought with Brian and Kevin maybe like 3,000. Kevin can comment more on that.
So we picked we looked at the 8 parents and we, obviously they're not segregating for all, 20 different QTL. They're only, in any one particular cross, they're only segregating for a subset of those. And so we designed the crosses so that each set of crosses were segregating for a different set of QTL. So I think, with several thousand, like we said, in the way that we set up these crosses, we should really be getting a large number of lines that have that will be fixed for most of the QTL. So back to the earlier question, the magic population wasn't really so much about mapping as it was about just finding a way to accumulate all of the potentially, favorable alleles in a reasonable malting background.
Okay, thanks.
Any other questions for Ahmed? And we'll...
Good work.
All right, I will share my screen and we can use the last few minutes to tap into all of your expertise. So let me share again. So obviously we don't want to deploy winter barley as we could deploy winter barley as itself, but we would like to deploy it as part of a winter summer annual cropping system. So we're looking at soybean as an obvious partner for that. So what we would like to do is to collect some data on that kind of a cropping system so that we can begin to characterize the potential value of a winter barley soybean cropping system. So this is some data from a study that was published by folks, a number of who are actually in the group today. So they could probably explain this, but I know they could explain this better than I could. But one of the things that we would like to do is look at cropping systems and look at nitrate leaching out of those systems. So this was a soybean system set up with these different cover crops and different treatments, and it shows that they were measuring nitrate levels with cup lysimeters at several depths in the spring and the fall in those different systems. And so what you can see is that the several of the winter cover crops, Pentecost, Camelina, rye did quite well in terms of reducing the amount of nitrates that were leaching out of that system. So I look at an experiment like this and I think, well, how can I want to create an experiment like this to collect similar data for barley. So you'll notice that rye, a cereal, was one of the treatments, and rye actually did quite well at reducing the amount of nitrates lost from the system. But in this experiment, as I understand it, the rye was actually winter kill or terminated. And then, because it did make it through the winter, was terminated, and then they planted soybean because the rye growing season is going to be too long to do a sort of a double crop or a relay system. And the camelina and pennycress systems were actually relay systems. So what can we do with winter barley? And so the overall objectives are, we want to compare the productivity of a single crop soybean system to a double cropping with winter barley system, and also compare nitrate in those systems to try to begin to characterize the potential value of those systems. So this is my attempt at a cartoon of a sort of relay type cropping system, variables that you could manipulate. So on the top is a single crop system. On the bottom is a true double crop system where you wait for the barley to be harvested before you plant soybeans. And we've done the experiment on the bottom to some extent. And Aaron Lorenz has looked at the impact of planting soybeans, much later than you normally would. And as we would expect, you get a much reduced yield when you do that kind of a system. The numbers I've put on the bottom are basically out of an extension page that Seth Nave has put together that shows that if you plant soybeans, this is just in a single crop system, if you plant soybeans by May 1st, you get 100% of the yield that you would expect. And then as you delay planting, you get reduced. This is what they would predict to be the reduced amount of yield you would get. And again, those are all in a single crop system. So that's one of the questions I think we have to figure out is how are we going to work this system? When would we plant soybeans into the barley to maximize the potential of this system? Because obviously, if you wait, too long, you're just going to lose yield because you're planting so late. And then, although we can look at sort of short season soybeans and other ways to mitigate that. But the earlier you plant, the longer the two crops are going to be in competition with one another, and that may have its own impacts as well. So that's sort of one of the issues I think that we have to really think about in the system. I mentioned that we were going to measure nitrates with this sort of cup lysimeter system that other people have used that we will be playing around with this summer. But I wanted to show you this slide because this is an experiment we planted last fall. As a little practice experiment, hoping that we might get funded to do something. So we have this experiment to work with this summer to see if we want to see how we want to design the full-blown experiment, which wouldn't get planted until fall. So what you see are just plots of, in this case, they're both wheat and barley. We left skip rows, so there's an empty row in between two rows of either wheat or barley, and then empty plots as well, so that we can compare a winter barley system with or a soybean alone system with a soybean planted into these skip rows of either winter wheat or winter barley. And so this is just a diagram of the plots and where we might place lysimeters at different depths. We would end up with soybeans that are 21-inch spacing in the system. So once the small grain is harvested, that's what the soybeans would be at, which is in the narrower range of row spacing of sort of standard soybean production, as I understand right now. And so this is the kind of experiment that I would envision planting in the fall in multiple locations. We have this one in St. Paul right now that we can play around with. And one of the things I'm thinking about doing is that these are planted as blocks. So there's 3 blocks here. I think one of the things I'll do is actually have each block be a different planting date. So we can start to look at the effect of the effect of planting the soybean into this cereal grain at different planting dates and how that affects competition while the two crops are both there, as well as harvest. So the other issue is if you plant soon and the soybeans get big enough, you don't want to take the tops of the soybeans off when you're harvesting the small grain. So I would be really interested in hearing suggestions from folks about things that we should, who have done this kind of experiment, about things that we should consider. I have talked to a few people about these experiments, but I would be really interested to hear either questions or suggestions from folks in this group about what we should be thinking about.
When will the winter barley be in the boot stage? Early boot.
Early boot.
Or don't even answer that in terms of when you could still get a certain type of soybean planter through the winter
I think we
barley
could go
without
into
doing
early
jamming.
May. I think we could go into early May.
I'm sorry.
1st of May, early May.
Yeah.
Um,
So that
Yeah.
would match up. So that would match up with potential soybean planting dates, right?
mhm. Yeah, so a May 1st planting date would be sort of a normalish, late-ish planting date. And I don't know how much into May we could go, but a little into early May. I haven't, yeah. It also depends. So we actually picked an early, it depends, there's wheat and barley in here too. So we picked a really early winter barley type out of our group, but the wheat will be quite a bit later.
So when will this, again, tell me, when will the winter barley be harvested?
We should be able to harvest the winter barley by the end of June.
So so the soybeans would be in for 2 months with the winter barley, right? 2 months.
Yeah.
Yeah.
Yeah, that would be probably the longer part of the window. If you could push it shorter than that, I don't know. So, yeah.
Yeah. So are there any other potential pieces of equipment that could plant later? Has anyone thought about
I have read, I don't know firsthand, but I have read some accounts of equipment that can sort of push the crop out of the way and seed into
Yeah,
something.
right.
But I wonder if anybody else has thoughts about that. I know Scotty's on here too, or was. For experimental purposes, we're going to use a push planter and hand plant these. But yes, I definitely want to be. I don't want to work in a system that's not going to be transferable to a commercial system.
All right.
I mean, we have the technology to plant into that. That's not a problem. I mean, there's it just would require resetting up our high clearance equipment to do it. I mean, it'd be drilled beans. I mean, my current planter, I mean, even at boot, I mean, that stuff is not gonna, we shouldn't break it even with our traditional planter either. It should be able to handle that quite well.
But could you get through it in June, on June 1?
So we went in and we did the opposite part of this. Alex and I did soybean maturity date and harvest timing on winter wheat planting. And we were able to get through the soybean with our equipment to plant the winter wheat. So I think we could do it. Again, the drill is I mean, that was not using the drill, that was using our high clearance equipment, but I think the drill could be set up to handle that just by removing units off of it. I mean, it's not currently set up it's set up, I think they're on 7-inch openings right now, but it could be. I mean, it would require probably the station a day or so to take the drill apart and set it up. But I mean, assuming you were going to go to a much larger, I mean, I wouldn't even bring it on campus for something like this, but if it was like a larger footprint, sure. I mean, I think where we were, using the drill for a day or two, I could see doing that.
So that would be my take in terms of start thinking about the type of commercial equipment that could be developed that would be able to plant into winter rye, like the first week in June.
Hey, Scotty, when did you guys harvest that pennycress last year in Rosemont again? Was that mid-June?
Yeah, you're talking about Soy Select.
Yeah.
That would have been, we were 3rd week of June.
3rd week of June, yeah. And we planted that right away, early May, maybe even 1st of May. So those soybeans were with that pennycress for almost 2 months, and they really looked good after the pennycress came off. So maybe the soybeans can stand in there for 2 months. I don't know.
Yeah, but I think pennycress is a bit of a different beast architecturally than a winter annual grain. So I think, but I mean, look, I guess the thing is we can give them, we have the technology to get the beans in the ground. I think that's number one. And I wouldn't even be afraid to drive in there, even with a traditional planter, even at, you know, even at or before boot. I don't think it would cause that much damage. I mean, we've seen big windstorms come down and push down some of our small grains right over there when we did the large rice study. That big storm came in and laid everything down, and it was standing back up, and some of that was already in anthesis. So I wouldn't worry too much about it. I think between the equipment on the station and between what we have in our inventory, we should be able to do it.
Carmen, what do you think?
Well, I was looking at it. Are you thinking of the soybeans? Would that be on 30-inch spacing then? Because you've got a skip row there. Just a wild idea. What if you did a row of barley, then a row of soybeans, row of barley instead of the 2 rows of barley? Would that make any difference on equipment movement through, or and so what you would be ending up is planting your soybeans on 15-inch rows, if I'm looking at this correctly?
Yeah, I think right now these would be, the soybeans would end up being 21-inch. So
Okay. Well, didn't Paul Porter do something with rye years ago, interseeded into standing rye with soybeans and didn't have any problem?
Well, he would have been doing dates of planting, and he went all the way up through what is called green planting, right? So it would have been when the rye would have been headed out, and then he killed it after planting, at the time of planting.
mhm.
Oh, I was under the impression that he had actually harvested rye and soybeans.
Well, I don't remember that. That could be, but I don't remember that.
Okay. Because I thought he cut the rye high enough so he didn't cut the soybeans off.
I don't remember that. It could be, but I don't remember that.
Okay.
I mean, for
So-
academic purposes, it might be nice to see, you know, if you could get in there and do a planting June 1st, but I don't think you would have to. I think if you had a early May, you know, around the 1st of May, mid-May, and maybe an earlier planting, say towards the end of April, I think that's going to give you a pretty good
Well, that's
experiment.
the case. We're home free.
Yeah. I mean, if the barley's coming off late June, yeah, your soybean, I mean, especially if you plant early to mid-May, your soybeans are still going to be small enough. Should be able to come over the top. And, you know, you got stripper headers, you've got ways of harvesting that barley and keeping the soybean down if the soybean does get some legs and is tall. You probably have better ways to harvest it than you do going in and planting the soybean, is what I'm saying. Well, I mean, there's equipment to do both.
So Aaron, in the study that you did with soybean and pennycress, were there issues at harvest when the soybean or when the pennycress was harvested in terms of impacting the soybeans?
No, the soybeans were fine. They cut high enough. The problem is they left a lot of pennycress out there, was the problem. And it's my understanding that the pennycress, for whatever reason, their pods, the seeds were lower down last year than normal. So there's a lot of pennycress left in the field. But
A lot of it was stunted in general across the entire network. I mean, based on all the winter annuals looked like they were shorter than what they should have been. And pennycress did exactly like that. And so at all our sites, you know, we were not dealing with the typical height that we would have. And then there were some delays with being able to get to the maturation that we needed to be able to harvest. Delays are really bad in a relay system because the beans keep going. As we dry down, light's coming through, and we just watch the beans take off. It was more of a problem in our camelina production system than anything else. I mean, at least with barley, all your material's up at the very top. So it's a little bit, probably should be a bit easier I mean, your competition's probably going to be a lot greater, but at least from a harvestability point of view, it should be a lot easier because your stuff's all up top.
And there could even be, you know, some architectural differences in terms of light penetration, that sort of thing that you could consider as well in terms of the choice of the barley variety, or even height for that matter.
Obviously, soybean variety would impact here. You've got some that are bushy in canopy and some that grow straighter. And so some of them wouldn't grow as tall as quickly.
Yeah, that's
Yeah.
true. Maybe we should pick out a bushy type and a marrow type.
Yeah.
You have those in sort of short season and regular season, or do you have some variation in that?
Yeah, we do. And then even commercial varieties are usually characterized for their bushiness.
So that's another variable, because the way we set it up is we'd have a couple of soybean varieties and a couple of barley, but we can change things, but that's what I would like to do, is play around with this practice experiment this summer to understand what's going on, and then by the time fall rolls around, you know what we want to do in terms of the design. So these comments are very helpful, and I'm jotting some of them down.
When was that barley planted?
Oh, does it say up here? Let's see.
Oh, maybe it's.
Cut that off. It was probably planted. I mean, this picture was taken in the fall.
Oh, okay.
And it was probably planted usually we sort of target the very end of September.
Okay.
But I can't remember offhand when we got this in.
Kevin, do you remember from last year when you planted soybeans after winter barley harvest? What did those soybeans yield? Do you remember?
Aaron, are we gonna ask? I don't know that we got a yield out of those. I could ask the station folks who harvested that field, but that was just I don't know if that was an MCIA field or a.
You're talking about the one on campus last year?
Yeah, the one that went in.
They must not have yielded much. They looked terrible. I mean, they made it, I think, but barely. They were really short. They couldn't have yielded very much at all.
And that was a straight up double cropping. Like we waited.
Yeah,
until,
that's what I mean. I was just wondering what it was, whether it was 10% of the yield or.
Oh, probably half.
Yeah.
Yeah, at the best, I'm guessing. They were really short.
I have some pictures of it, but as I recall too, the rows didn't really fill in. We probably could have planted them a lot denser than that, and it would have had a better yield. I mean, that's something to think about too.
That's true. That's true.
And when you said half, you mean half of
what they normally would.
Well, that's not bad. What's wrong with that? You got. Well, you're just.
Depends how much the winter barley is worth.
You got to give credit to the barley, man.
Yeah.
Aaron,
that's what I'm just starting to think about, in terms of total cropping system yield.
Right.
What did that look like?
I mean, that's another part of this, right? So this experiment is set up so we're going to measure the yield out of both components, and then also, do quality on them to sort of see what the value is. Because if you get, real good malting quality price and whatever the soybean price is, even if the yields aren't
that's what I mean.
high, you could still make it a profitable system, I think.
Aaron, you have to recognize that grains have value too.
I guess so.
I like to think of this as a winter barley crop with a soybean cover crop.
Relegated to a cover crop breeder. Just kidding. We're also going to put some specialty soybean varieties in here as well that may command some sort of premium. So
that sounds like a good idea. Yeah.
Maybe that would impact the economics in a favorable way.
Aaron, I had a question. You said 50% reduction on the double crop, but in those in your pennycress experiment with the relay crop, do you have an estimate on the reduction there on average?
Geez, you put me in the spot in terms of an actual number, but it wasn't nearly that much. It was maybe 10%.
Okay.
Yeah, it wasn't too much. I was actually really impressed with how well the
Okay.
soybeans did in that system. And so having done both of these systems now in the short time I've been here, I'm definitely leaning towards the intercropping system because the soybeans really got hit really hard in that double cropping, really late planting scenario.
Yeah, but be careful though, you know, a half of a soybean yield plus the winter barley. I mean, that someone needs to look at the economics of that though.
Yeah, it could be there, but I think we can get even more if we intercrop it.
No. I know, but I'm just saying in terms of just blowing that away, right?
Yeah.
And also putting in higher value soybeans into the system, right? So I wouldn't throw that idea away.
Okay.
So Aaron, would you expect there to be any like penalty on the quality of soybean coming out of this by either the competition of being in the intercrop or the late planting?
In terms of protein oil?
Yeah.
No, the protein
Anything that
oil,
would affect the value.
in the pennycrust experiment last year, the protein oil on average was really similar. I mean, there was hardly any difference there at all.
Okay.
Yeah, there's no effect there. And on the double cropping side, there was a small effect, but not much. I have to go back and look at Becky's thesis to know for sure.
Okay. I guess I can do that too.
Can I switch gears and ask if you've done any interseeding and standing corn? I'm looking at the late possibilities here of planting the barley. And, you know, a lot of people are putting rye into standing corn. I'm wondering about barley into standing corn October 1. And then thinking in terms of planting in that corn residue, would there be better potential for winter survival?
Yeah, those are great questions. I mean, one of the conundrum with corn is that it has great stubble for trapping snow and winter survival potential, but it's also a great source of inoculum for Fusarium head blight, which is the problem with barley. So there's a little bit of a double-edged sword there, but it's an interesting idea. And I mean, one of the things that we're curious about with winter barley too is whether or not that earlier growing season would actually help avoid some of the diseases that are more of a problem in spring barley, and scab might be one of those.
Good. Okay.
Are there any questions from our friends in Wisconsin?
I got to see who the friends in Wisconsin are. Okay.
If not, well, to everybody, thanks for the great update, great discussion, and I think you guys are making good progress in moving the winter barley forward, and I think this network of breeding and genomics work that you guys are doing, I think could very well move it forward in a reasonable period of time. So I'd like to congratulate you guys for putting a great team together.
Well, thanks, and thanks for all of the suggestions. I may come back and tap some of you later on as the summer goes on and we see what happens with this pilot experiment.
Okay. So thanks everyone for joining us today. And next week, I believe we're getting an update from Lee DeHaan's genomics and breeding program at the Land Institute.
Oh, awesome.
I think that's the upcoming conversation next week, if I remember correctly. So take care, everybody. Stay well and have a good few days off. So
thanks.
Yeah. Bye-bye.
Thanks, Kevin.